All right, today we're going to tackle human African trypanosomiasis, a disease you probably know better as sleeping sickness. This is a really fascinating story about a tiny, shape-shifting parasite, its accomplice, the tsetse fly, and a disease that plays out in two completely different ways across the African continent. So let's jump right in. So this is the big question, isn't it? How can a single-celled organism, this tiny parasite, manage to outsmart our incredibly complex immune system? Not just for a few days, but for months, or in some cases, for years. How does it pull off this incredible disappearing act? And what's the ultimate price for the person who gets infected? Okay, so here's how we're going to break it down. First, we're going to get to know the parasite itself. Then, we'll trace its pretty wild journey between two different hosts. After that, we'll dig into why the disease looks so different in West Africa versus East Africa, uncover how it attacks the body and evades our defenses, and finally, look at how we try to fight back. Let's start with the main character in our story, the parasite, Trypanosoma brucei. Now, the first thing you really need to grasp about this organism is that it's an extracellular parasite. And that's a huge deal. It means it doesn't bother hiding inside our cells. Nope, it lives right out in the open, swimming around in our blood and our lymph fluid. And to get there, it needs a getaway driver, which is where its partner in crime, the tsetse fly, comes in. To really get what makes this parasite so successful, you have to follow its remarkable journey. It has this complex two-host life cycle, and it's constantly shape-shifting as it moves between an insect and a human. Let's trace that path right now. So, it all kicks off with a bite from an infected tsetse fly. It injects the parasite in one form, and once it's inside you, it transforms and starts multiplying like crazy in your blood. Then, a new, uninfected fly comes along, bites you, and sucks up some of those parasites. Inside the fly, they transform again, make their way to the salivary glands, and morph into the infectious stage, all set to start the whole cycle over in a new person. This constant shape-shifting is the absolute core of its survival strategy. Now, here's where it gets really interesting. Trypanosoma brucei isn't just one thing. It actually has two major subspecies, and they cause two dramatically different diseases in different parts of Africa. Over in West and Central Africa, you've got Trypanosoma brucei Gambian. This is the one that causes the West African version of the disease. But then in Eastern and Southern Africa, you find Trypanosoma brucei Rhodesian, and that causes the Eastern form. We're talking about the same parent species here, but these two subspecies lead to two very, very different outcomes for the patient. And this split isn't just about names. It's also tied to the land itself. TB Gambian prefers the lush riverine areas of West and Central Africa because it's carried by tsetse flies that live near water. On the flip side, TB Rhodesian is a creature of the savanna, transmitted by flies you find in the dry grasslands of Eastern Africa. And this right here, this is probably the single most critical difference between them. The West African form is almost exclusively a human disease. It gets passed from person to person by the fly. What does that mean? It means it causes a chronic illness that can just simmer away for years. The East African form, though, is zoonotic. Its main home is in wild animals and cattle. So when it spills over into humans, it causes an acute, fast, and aggressive illness that is often fatal within a year, sometimes even before the classic neurological symptoms have a chance to show up. So what does this disease actually look like for a patient? And more importantly, how does the parasite manage to hang around for so long, especially in that chronic West African form? Let's focus on the chronic Gambian form. The progression is just so slow and insidious. It might start with a sore at the bite site. Then comes stage 1, where the parasite is in the blood, causing fevers and swollen lymph nodes, and there's this classic sign called winter bottom sign, a telltale swelling of the lymph nodes in the back of the neck. But the real turning point is stage 2. This can be months or even years down the line. It's when the parasite breaches the blood-brain barrier and invades the central nervous system. And that's when you see the sleeping sickness symptoms: confusion, apathy, that classic messed up sleep cycle, which, if it goes untreated, leads to coma and eventually death. So how on earth does it survive in the body for that long? The answer is this absolutely brilliant, almost diabolical strategy called antigenic variation. Think of it like this. The parasite wears a uniform, a dense coat made of a single protein.
Our immune system spots this uniform and starts making antibodies that are perfectly designed to destroy it. But right when our immune response is about to win, the parasite does something amazing. It just changes its uniform. It switches to a completely different protein code from a massive genetic library it has. All of a sudden, our custom-made antibodies are useless. The parasite population roars back, and the whole cycle of fever and remission starts all over again. It's an incredible bait and switch.
Okay, so we've seen the parasite's playbook. Now let's talk about ours. How do we actually diagnose, treat, and try to control this incredibly tricky disease? Well, for a definitive diagnosis, you have to actually find the parasite. The most direct way is to just look for it under a microscope. You can take a sample from a swollen lymph node in stage 1, for example. But to confirm stage 2, you have to do a lumbar puncture to get a sample of cerebrospinal fluid. That's the only way to know if it's in the brain. And for those really tough cases where parasites are hard to find, we have more sensitive methods, like PCR.
Now, treatment is really complicated. It all depends on two things: which subspecies it is and what stage the disease is in. In the early stage, before it's in the central nervous system, we have drugs like pentamidine and suramin. But the big problem is that those drugs can't cross the blood-brain barrier. So for late-stage disease, for decades, the main option was a drug called melarsoprol. And get this, it's an arsenic-based compound. It's incredibly toxic, and it can be fatal itself. But it was used because it was one of the few things that could actually get into the brain to kill the parasite.
So ultimately, when you zoom out, controlling this disease really comes down to a three-pronged attack. First, you have to break the cycle by finding and treating sick people. Second, you have to go after the vector itself, controlling the tsetse fly populations. And third, you use public health strategies to try to keep people and flies away from each other as much as possible. Which really leaves us with this final, challenging question. You've got a parasite that can change its disguise an almost infinite number of times, and in one of its forms, it has this huge reservoir in animals that we can't easily control. So, can a foe this adaptable, this clever, ever really be eradicated? That's the question that public health officials and researchers are still grappling with today.